I will give enough time to write your note. Therefore, initially, you can hear me, listen me. Then, yes, our topic is nucleic acids. Our topic is nucleic acids. They are polymers. You know, they are polymers. Exist as polynucleotides. As they are polymers. In the case of nucleic acids, we name these polymers as polynucleotides, made up of monomers called nucleotides. Everything you know. They contain CHO and P. Nucleic acids are macromolecules. Macromolecules and again polymers. Right? As they are included in biological systems, both humans, animals, plants as well, and some microbes, never. They are included, they are biological systems, DNA and RNA, they are biopolymers. Biological polymers mean biopolymers. There are two types, DNA, that means deoxyribonucleic acids, and RNA, ribonucleic acids. And what about the structure of nucleotides? Again, you have to draw this one. No need to remember this chemical nature. Anyway, you can take a screenshot and stick to your note. You can take a screenshot now. Okay, then you know it has three components. Pentose sugar. It should not pentose sugar group. And it should pentose sugar. Nitrogenous base. This is the structure of nitrogenous base and the phosphate group. A nucleotide without a phosphate group is called a nucleoside. Without phosphate group, this group we called as nucleoside. Nucleoside. And after adding phosphate group, the whole structure we called as the nucleotide. What is the difference between nucleotide and nucleoside? Tell me.
Nucleotides have a phosphate group and nucleoside doesn't have a phosphate group.
Very good. Examples for nucleosides: adenosine and guanosine. And pentose sugar. Pentose sugars are two types, namely deoxyribose. In deoxyribose 1, oxygen atom is less than ribose. You know. Nitrogenous bases, there are two types of, two major groups of nitrogenous bases, purines and pyrimidines. Purines are larger in size with 2 rings. Pyrimidines smaller in size with a single ring. In purines, there are two types, adenine and guanine. Pyrimidines, 3 types, thymine, uracil, and cytosine. Bases are commonly represented by letters A, G, T, U, and C respectively. Phosphate group, it gives the nucleic acids the acidic nature. Yes, these things you have written or not up to now, last week.
Written, teacher.
Written or not?
Have written, teacher.
Then formation of nucleic acids. Today our topic actually formation of nucleic acids. And finally we have to discuss about the Watson-Crick model of DNA. Formation of nucleic acids, millions of nucleic acids, nucleotides actually, millions of nucleotides like ribonucleotides and deoxyribonucleotides joined by phosphodiester bond to form polynucleotide nucleotide chains by condensation between OH group of the phosphate group of one nucleotide with the OH attached to the third carbon of pentose group of the other. By the condensation reaction between these two, they form phosphodiester bonds. These bonds results in a backbone with the repeating pattern of sugar phosphate units. Nucleic acids are linear polymers of nucleotides. That means we have discussed in some cyclic structure in carbohydrates, no? When we after adding monosaccharides into water, they form cyclic structure. Anyway, they never form cyclic structure. Always they are linear ones. Always nucleic acids are linear polymers. There are two kinds of nucleic acids depend on the type of the sugar molecule involved. If the sugar molecule in the nucleotide is deoxyribose, the nucleic acid is DNA. If the pentose sugar is ribose, then the nucleic acid is RNA. DNA contains adenine, thymine, guanine, and cytosine. RNA contains adenine, guanine, cytosine, and the uracil. These things you have written or not?
Written, teacher.
Yes. Then structure of DNA molecule according to the
Noted.
Watson-Crick model. Why we call this structure as Watson and Crick? This was introduced by Watson and Crick. And the two scientists, Watson and Crick. DNA molecules have two antiparallel polynucleotide chains that spiral around an imaginary axis, forming a double helix. Around an imaginary axis means imagine a helical staircase. Are there any axis, axial things at the center? Are there any real axis?
No, teacher.
Axis. That means if they have an axis like this, it is rotated around this axis like this, but no any axis. We imagine there is an axis and the DNA molecule is actually it is winding around this axis. No actual axis at the center. Now is it clear?
Sometimes it gets stuck, teacher.
Yes.
Sometimes your voice gets stuck. Now, okay, teacher.
Yes, that means no actual axis at the center. No actual axis at the center. The DNA molecule is winding around an axis. Actually, we can't see an axis at the center. We imagine there is an axis. That is why we called as it is an imaginary axis. No actual axis, but we assume. Can you see the screen?
Yes, teacher.
Yes. DNA molecules have two antiparallel polynucleotide chains that spiral around an imaginary axis, forming a double helix. The two sugar phosphate backbones run in opposite direction from each other, opposite forms of each other, and the arrangement is referred to as antiparallel. Why? One strand appears in 3 to 5 direction, other one is 5 to 3 direction. No? First one is 3 to 5, other one is 5 to 3. That is why we called as antiparallel. Antiparallel. The sugar phosphate backbones are on the outside of the helix. You know the sugar phosphate backbones in this ladder, the outside lines are represented by two sugar phosphate backbones. Which components are included in the sugar phosphate backbone?
Phosphate group and pentose sugar.
In all nucleotides which are involved in the formation of polynucleotide, all the pentose sugars and all the phosphate groups makes the sugar phosphate backbone. Outside lines are represented by sugar phosphate backbones of each two strands. And the nitrogenous bases are paired in the interior of the helix, like that. Nitrogenous bases and hydrogen bonds. Those are the interior of the helix. Those are the interior of the helix. The two strands are held together by hydrogen bonds between the paired nitrogenous bases. We know base pair rule. Always purine base pairs with the specific pyrimidine base. You know, adenine, guanine, and yes, these two are purine bases. These two are pyrimidine bases. Always purine base pairs with the specific pyrimidine base. You can complete the note initially, this part.